In this video, we're going to talk about transcription as it occurs in eukaryotic cells. And let's take a look at some differences between transcription in eukaryotes and transcription in prokaryotes. In prokaryotes, transcription occurs just within the cell itself. There is no nucleus, and so transcription occurs in the same place that translation occurs. And so what we'll learn a little bit later is that as the DNA is being transcribed, right, so here's our messenger RNA being made from the DNA, even before that messenger RNA is done, ribosomes are jumping onto that messenger RNA and making protein from it. Whereas in eukaryotes, because there's a nucleus, the location of transcription and translation are different. Transcription occurs within the nucleus, and that's where the messenger RNA is made and processed. It's then exported out of the nucleus to the cytoplasm, where it's then a substrate for translation. Now, if you compare the RNA processing between eukaryotes and prokaryotes, you'll see that the processing is much more extensive in eukaryotes. In prokaryotes, there's a little bit of processing of tRNAs and ribosomal RNAs, but there's really no processing of messenger RNAs, and that makes sense because right as soon as that messenger RNA is being made, it's a substrate for translation, which means there's really no time for it to be processed. But in eukaryotes, there's a lot of processing that occurs within the nucleus before it is transported to the cytoplasm. We have a 5' cap that's added to the RNA, a poly A tail, there's a bunch of introns that need to be spliced out. So there's a bunch of processing that we will look at in the next lecture. Now, prokaryotes have just one RNA polymerase, whereas eukaryotes have 3, RNAP1, 2, and 3. Now, the one that we're going to be focusing on is RNA polymerase 2, which is mostly involved in transcribing the genes that give rise to messenger RNAs that will then eventually give rise to proteins. Now, the first step of transcription in eukaryotes is the pre-initiation complex. And to understand that, we have to understand here a promoter in eukaryotes. We have our initiator sequence. This is the sequence that DNA is going to unwind and is going to be the start of transcription. The TATA box here is a portion of the promoter to which a very specific general transcription factor is going to bind, the TATA binding protein. Now, with later lectures, we'll talk about enhancers, and enhancers are sequences of DNA that help regulate exactly when and in which cell a particular gene is going to be expressed. Now, remember that in RNA polymerase in prokaryotes, there was that sigma factor. Right? It was separate from RNA polymerase, but it worked with RNA polymerase to bind to the promoter. Now, the evolutionarily similar or analogous components of eukaryotes that act like a sigma factor are called general transcription factors. Now, I don't have the word general here, but it is pretty important because we're going to talk about specific transcription factors later on. Okay, so general transcription factors are these here. So the TATA binding protein, like we already mentioned, TF2A, B, E, F, and H. And then there's also a TF2D, which the TATA binding protein is part of. And all of these bind to various regions within the promoter and then help recruit the RNA polymerase to that promoter. So let's go through the steps necessary to start an initiation complex. And the first is that TF2, so that stands for transcription factor for RNA polymerase 2D. So TF2D, along with the TATA binding protein, bind to the TATA box of the promoter. And then they recruit TF2A, which then helps stabilize the binding of TF2B. Now, this whole complex then goes on to recruit, finally, RNA polymerase. that comes with TF2F. Right, so now what we have here is we've got our RNA polymerase that isn't really directly binding to the promoter. Notice it's sort of being recruited there by all of these general transcription factors. Then TF2E binds and recruits TF2H. So TF2H is one of the most important of these general transcription factors in that it has kinase ability, which means it can phosphorylate things, and it has helicase ability, which means it can separate and unwind the DNA. And it does so right here at the initiator, and that's forming the open complex. So we've got our initiator complex here, or our initiation complex here, and we've actually catalyzed the opening of the two strands of DNA. And so again, this is called the open complex. Now notice RNA polymerase has this tail. This is the carboxy terminal domain of RBP1. It's one of the subunits of the eukaryotic RNA polymerase II. And notice, look at the similarity between that tail there and the carboxy terminal domain of subunit alpha in prokaryotes. The difference here is, remember, the alpha carboxyl terminal domain in prokaryotes physically binds to the up element, that part of that third site of contact with the promoter. Well, this tail here, this carboxy terminal domain of RBP1, doesn't interact directly with any DNA, but instead interacts with the various general transcription factors, right? Holding that and acting as another site of contact then to this general area, to this promoter. Okay, at that point though, because of that contact, the RNA polymerase is not going to be able to start elongation. So in order to clear that promoter, what happens is TF2H, if you'll recall, it has kinase activity. TF2H phosphorylates that carboxyl terminal domain of RBP1. And so look at all that phosphorylation on that carboxy terminal domain, and that now inhibits any interaction with the general transcription factors, and that allows the RNA polymerase to finally clear the promoter and begin elongation. And the only thing that kind of sticks with this is TF2F stays associated all the way through elongation. Now, as elongation is occurring, there's several things that are happening. First, they recruit these elongation factors, and these are proteins that keep that RNA polymerase moving, keep it from stalling. In addition, we have various molecules are going to be phosphorylating this RBP1, the carboxy terminal domain, in various ways. And that sets up a code. Okay, so again, here's that carboxy terminal domain, here's that tail. And it's going to be, as elongation occurs, it's going to be phosphorylated in different patterns. We have a pattern here, we have another pattern here, we have another pattern here. And these patterns are recognized by very specific enzymes that have to do things at very specific times during transcription. So for example, this first pattern of phosphorylation here on the carboxy terminal domain of RBP1 attracts a capping enzyme. Now we're going to talk about this in the next lecture, but what this does is it adds a G-cap to the 5' end of, whoops, not here, of the emerging messenger RNA. And then we get another pattern here. After this has been completed, more kinases, more phosphatases come in, they change the phosphorylation pattern, and that attracts the various components of the splicing machinery that kind of sit and wait for introns to be made, and then they're going to splice them out and splice the exons together. Now once all that is completed, so this is while transcription is occurring, the phosphorylation pattern again changes for the last time. That attracts the polyadenylation and the cleavage factors, that when that particular sequence that we'll talk about soon emerges, that cleavage sequence emerges, these proteins will bind to it, cut that messenger RNA, and end the transcription. In addition, and we're not going to go into the details, so don't worry about trying to memorize any of these proteins here, but don't forget that DNA is in the form of nucleosomes. It's got that DNA wrapped around that histone octamer, and so those have to constantly be disassembled and then reassembled as that RNA polymerase moves down that DNA as it's transcribing. So it's pretty complex. Now, at some point in time during the elongation, this sequence here, what's called the cleavage signal sequence, is transcribed from the template strand here. So here's the template strand, and here it has been transcribed into messenger RNA. Now that is going to attract the polyadenylation factors, and it's going to attract an endonuclease that cuts just past that cleavage signal sequence. Okay. And then the polyadenylation factors that are bound to this sequence then will then add a poly A tail. Again, we'll get into more details about that kind of modification in another lecture. Now, what we have here is after the nuclease cut the RNA, we have a 5' end of an RNA that doesn't have a cap on it. Remember, because this has already been capped here. But when you cut it, the new 5' end that begins to emerge is uncapped, and there is a exonuclease that recognizes that and chews it away and knocks off the RNA polymerase from the template. Okay, so that concludes our discussion of transcription. The next lecture we're going to discuss all the processing that we sort of briefly mentioned, how that messenger RNA is processed during transcription, the poly A tail, the 5' G cap, and the splicing out of the introns and the splicing together of exons.